DNA replication and cell division. The ability of DNA to make a copy of itself. We call this DNA replication. This is the basis for reproduction and inheritance. DNA molecule exists within chromosome, that is in the nucleus, and is surrounded by a soup of free DNA nucleotides, which is to build new DNA molecules. DNA structure is double helix and must replicate semi-conservatively. And replication is called semi-conservative because each new double helix is composed of an old, that is parental, and a new, that is daughter strand. Here is just a soup, soup. So it says DNA molecule exists in chromosome, which is surrounded by a soup of free DNA nucleotides, which is important during DNA replication, because DNA is built from the monomers of what? Nucleotides. So the soup of nucleotides, free nucleotides, will be brought by enzymes and then added to the 3' end of the growing strand of the new DNA or new DNA strand. Here is nucleotide. Nucleotide is made up of the 3 components. Sugar, that is 5-carbon sugar, nitrogenous base, and phosphate. This is the DNA structure, double helix structure, 5' end to 3' end. In this case, from this direction, 5' to 3' end. So this N indicates, okay, N's are here, N around the DNA structure. That is, I want to show you as a soup of DNA, as a soup of nucleotides. So these nucleotides will be important during DNA replication. Therefore, let's say this one strand will be replicated during the DNA replication. This one strand will help us to form the new strand. So from 5' to 3', this one, as this end it is 5', so consider this is that strand, and the new DNA strand will be made from 5' end from this direction to this. In this case, the nucleotides from this soup around the DNA will be brought and added, and then the DNA will grow to this direction. The same is true for the other strand. So this strand, as the 5' end from this direction, will be template, and then the new strand from this will be made from 5' end from this direction. Then the strand which will made from this strand will be exactly the same with the previous other strand, because there is complementarity of the nitrogenous bases or the nucleotides. The nucleotides based on the nitrogenous bases, that is the adenine pair with thymine, guanine pair with cytosine. Mechanism of DNA replication. This is what? Semi-conservative. The proposed models were conservative way of replication, dispersive, and semi-conservative. And the accepted one is semi-conservative. What is each kind? What is conservative means, dispersive means, and semi-conservative means. Consider this is the original DNA molecule. This one is original, the blue color. In the first DNA copying or replication, in the case of conservative, this one is the old double-stranded DNA, and this one is the new. The two strands are together, the old one are together here, and the new one are separately together here. Then this is we call conservative. And in this case of dispersive case, here you can see this is one is the blue color is old. So here is the old strand, the fragment of old strand, then the new strand come, then the old strand, then the new strand, old strand, new strand, okay, in the fragmented way. That is dispersive. The same is true here. In the semi-conservative way, here from the daughter strands, this one are the daughter strands, one is old, the blue color is old, the other one strand is new. The other here, one strand used as a template, and then there will be the new strand here. That is the accepted one, the semi-conservative one, is the way of DNA replication. Several enzymes are involved in this process, and the main are in the DNA replication process. There are several enzymes. The molecular tools are enzymes you can consider in every reaction or every process within biological molecules or in organisms. The tools are enzymes. The same is true in this case. There are different enzymes. Just only the general enzymes or the general tools we will see are the DNA helicase and the DNA polymerase. There are other enzymes in the process. What is DNA helicase do is it breaks hydrogen bonds to ravel two single strands and unwind or open the helix DNA. The DNA replication will not take place as the helix is there. It should be opened and unzipped. Okay, it should be unzipped. This unzipping or opening the twisted DNA structure is by the enzyme of helicase. DNA polymerase, from its name, poly means many, so it makes what? The DNA will be having many nucleotides. Okay, by bringing the nucleotides from the soup of the nucleotides, then it will make many nucleotides per DNA. So its function is that. So DNA polymerase follows the helicase enzyme along each strand, which acts as a template for the synthesis of new strand. DNA polymerase assembles free DNA nucleotides into new strands alongside each of the template strands. The base sequence in each of these new strands is complementary to its template strand because of base pairing group. That is adenine always with thymine and cytosine always with guanine. Therefore, if you know one strand, then you can easily draw the other strand. DNA replication is this is the figure to show you the DNA replication. This is the original DNA, double-stranded DNA. When it is replicating, it will be unzipped and opened like this. Then the new strands will be made here. So one strand here is continuously moving without gap, and the other strand you can see is there is gap. This is because of the chemical direction of the DNA. In this picture you can easily understand that. Here is helicase, the enzyme helicase, which is opening the DNA to this direction, and this old strand is 5 prime. Okay, yeah, here the growing strand of the new growing strand of the DNA is 5 to the 3 prime direction at this end. Then the polymerase can simply add the nucleotides at 3 prime end. Okay, that is why there is no breakage of the new strand here. In this case, you can see here the new strand is broken strands. There are the fragments. These are we call these fragments Okazaki fragments. This is the scientist name who found these fragments. Then we call this breaking or fragmentary strand, lagging strand, and this unbroken or continuous strand, the leading strand. So here the direction of replication is to this direction, and the new strand which is towards the direction of replication is the leading strand, and the strand away from the direction of the replication is lagging strand. So here these are the DNA polymerases which are bringing the nucleotides and adding to the growing chain of the DNA. Then cell division. DNA replication and cell division are in one group or in one category because in each and every cell division there should be DNA replication. Cell division is a process by which a parent cell divides into two or more daughter cells. It usually occurs as part of a larger cell cycle. In eukaryotes, there are two distinct types of cell division. Vegetative division that we call mitosis, whereby each daughter cell is genetically identical to the parent cell. And the other kind of cell division is reproductive cell division, which is important for reproduction of organisms, whereby the number of chromosomes in the daughter cells is reduced by half to produce haploid gametes. This we call the process of meiosis. Mitosis, simple diagram. Here is consider this is one cell containing the DNA, the genetic material. Before this cell division, there should be the division of genetic material. DNA divide first, then cell will be divided into two, then DNA divide, then the cell will divide. Then the process will continue in such fashion, which is very important to maintain the genetic similarity between parental cell and the daughter cells. Meiosis results in four haploid daughter cells by undergoing one round of DNA replication followed by two cellular divisions. So there will be four daughter cells from male parent and also four daughter cells from female parents. Homologous chromosomes are separated in the first division. There are two consecutive divisions in meiosis. So in the first division, there will be homologous chromosomal separation. And sister chromatids will be separated in the second part. Both of these cell division cycles are used in the process of sexual reproduction at some point in their life cycle. Both are believed to be present in the last eukaryotic common ancestor, that is, which passes through generation to generation and just it exists today. Prokaryotes, mainly prokaryotes are bacteria, undergo a vegetative cell division known as binary fission, a kind of mitotic cell division. Specifically, we call binary fission, where their genetic material is segregated equally into two daughter cells. Bacterial cell division is just dividing into two, and continue like that. While binary fission may be the means of division by most prokaryotes, there are alternative manners of division, such as budding, that have been observed in eukaryotes. All cell divisions, regardless of organism, either prokaryote or eukaryote, all cell divisions are preceded by a single round of DNA replication. There should be DNA replication before cellular division. This is a binary fission. This is a bacteria. Before this bacteria divided into two, you can see here is the division of the genetic material. This is the genetic material, that is chromosome. Then it is going to divide it into two. Then in this phase it is divided into two. Then cell is going to divide. Finally, there will be cellular division. So the genetic material is found in both cells. This is a kind of bedding. This is parental hydra, a kind of organism. Here you can see cell will be when you remove this part, and then this part will be considered as one organism finally. So this is considered, this comes from in such way like this. Then this will be the parent for the next daughters. So parental hydra or parent hydra, then developing bed, this is bed, then new bed is forming, then finally it will be new organism or new hydra. For simple unicellular microorganisms such as amoeba, one cell division is equivalent to reproduction. Mitosis is important in such organisms that it is a means of reproduction. Therefore, an entire new organism is created. On a larger scale, mitotic cell division can create progeny from a multicellular organisms, such as plants that grow from cuttings. So mitosis is also important in plants for reproduction in such a way. For example, you can consider sugar cane, sunflowers, flowering plants can be reproduced in such a way. Mitotic cell division enables sexually reproducing organisms to develop from the one cell zygote. From zygote, during fertilization, the cell is one celled, which itself was produced by mitotic cell division from gametes. I mean mitotic cell division by from gametes. So gamete formation is what? Mitotic cell division. So mitotic cell division, then zygote formation, then zygote will be there will be mitotic cell division in zygote, then that will become multicellular from one cell to multicellular by mitosis. After growth, cell division by mitosis allows for continual construction and repair of the organism. You can consider zygote will be finally an individual or an organism. The human body experiences about 10 quadrillion cell divisions in a lifetime. You can think this number, very huge number. Okay, so in lifetime there will be such a number of cell division. The primary concern of cell division is the maintenance of the original cell's genome. Okay, cell division is very important that it will maintain the original cell's genome. What is genome? It is the total or whole content of the genetic material in an organism or in a species. Before division can occur, the genomic information that is stored in chromosomes must be replicated or divided, and the duplicated genome must be separated cleanly between cells. A great deal of cellular infrastructure is involved in keeping genome information consistent between generations. So, through this process, there will be genetic information consistent. There is no variation from generation to generation because of this process. These are the major types of cell division: binary fission, mitosis, and meiosis. So binary division, as we have seen in the bacteria, then there will be, this one is the genetic material, this one is the whole cell, the whole organism, considered as a bacterium, and here is the cell, the genetic material replication or DNA replication. Then first DNA replicated, then cell will be divided into two. Mitosis, just the cellular division, diploid cell divide into two. There are different phases. The phase of cellular, the genetic material division within a cell is called S phase or synthesis phase. Then finally there will be division of the genetic material. Then there will be division of finally the cell. Then meiosis, there are two phases. The homologous chromosomes separate at the first phase, then cell divide. Then finally the chromatids will be separated, and then the next cell division. So there are two consecutive cellular divisions in the meiosis. Then in the meiotic cell division, there will be gamete formation. When gametes come together, then it will form zygote. Thanks for watching.